we get started. So today's not for points, no names required, just want to get some feedback. We've moved from sex chromosomes into the central dogma. So the first thing we need to talk about is the gene. The gene is the first step, DNA, the first step of the central dogma. And before we get there, though, I said I was going to leave some time for questions about chi-square analysis, especially if anybody has attempted some or all of those Google Sheet exercises that I provided. Got time to answer questions if there are any. Okay, maybe Monday. I hope. One of the practice questions for today was to look up the name of the gene that initiates sex determination. We talked about this last class, actually, so this wasn't even looking up. What's the name of the gene that does sex determination in humans? SRY. We talked about that last class. That is the one gene that's on the Y chromosome in males that produces male development. In other words, if an individual does not have that gene, what sex, genetic sex would they be? If an individual doesn't have SRY, like let's say there was an X chromosome and the Y chromosome, which we know is not Y-shaped, it's just a smaller, fallen apart version of the X chromosome. But it normally contains this gene, SRY. What happens if you have a Y chromosome, but that gene is missing? What sex do you develop as? Female, because there's no gene that starts male development. SRY, that's its job, that's its role, its function. It's to turn on male development. So even if you've got a Y chromosome, if you look at the chromosomes and you say, oh, there's clearly a heteromorphic pair, differently sized pair, and this would have to be, the X and the Y would have to come from what? Normal phenotypic sex, as opposed to ZW. XY is in what sex? In males. If we were talking about the same type of chromosomes, but we were talking about chromosomes that had come from a female, phenotypically female individual, then that would be which chromosomes? Female has heteromorphic sex chromosomes. These are not XY. These are Z and W. Which one is the sex chromosome? Which is the one that causes femaleness? The W or the Z? It's ZW heteromorphy. So it's the female has the W chromosome. That is the female determining chromosome. So it's the equivalent of the Y chromosome. It's a gene that instead of carrying a gene that turns on male development, it carries a gene, or it contains a gene that is female determining, female symbol and then determining, female determining gene. Is on the W chromosome. So, quick review from last time. But then this question goes on. Beyond just knowing the name of the gene, this chapter, talking about the gene, we're going to look at DNA sequences of genes, how to get the DNA sequences of genes from online databases to start. So, I'm not going to ask you to tell me what the DNA sequence of this gene is, because how would we do that? I guess you could read out a DNA sequence, but that would be boring. Where's the gene located? Probably already know the answer to that question, too. It's in the human genome. It's on a particular chromosome, the Y chromosome. What makes this gene unusual compared to most other human genes? Open question. What do you think? Did anybody investigate this at all? Or do you know something about SRY that you'd like to share? Well, there's one thing that's weird about it drawn up here that we could start with. The X and the Y are homologs. For all other chromosome pairs, what's true about the two homologs? If there's a gene on one, it's on the other copy. That's not true of most genes on sex chromosomes, at least in the human case, the X and the Y. SRY is only on the Y. There's no SRY on the X chromosome. That's kind of weird. That's typical of most sex chromosomes, not just in humans, but in general. A lot of sex chromosomes, you'll find specific genes on the sex chromosome, the Y, for example, but not on the X. So SRY really is on the Y chromosome, and it's Y limited, or it's unique to the Y chromosome. Because what would happen if SRY got on the X chromosome? What effect would that have? If there was a copy of SRY on the X, then would you have it?
Yeah.
I'd have it. You'd have it. Everybody in the room would have it because we all have X chromosomes. Right? Females have X chromosomes, males have X chromosomes. So if SRY got onto the X chromosome, then it wouldn't be a sex-determining gene anymore. Everybody would be male because everybody would have SRY, and then we'd be extinct because there'd be no more sexual reproduction. So by necessity, SRY is only on the Y chromosome. If it moved to another chromosome, this is just an aside, hopefully it's interesting. If the SRY gene moved to a different chromosome, like chromosome 22, then we would call chromosome 22 the Y chromosome. Whatever chromosome has the male determining gene, the sex determining gene, is by definition the sex chromosome. And genes do move around from chromosome to chromosome. It's kind of weird. We'll talk about that hopefully in a week or so. We talk about chromosomal rearrangements. But genes do move from time to time from chromosome to chromosome. Over long periods of time, it's not rapid. But they do get around. And especially sex-determining genes tend to hop from chromosome to chromosome, which is kind of odd. What type of a protein is encoded by SRY? If you Google SRY or do some sort of web search, we can find out what sort of a gene it is. That's a topic we're going to be talking about a lot in the central dogma and the next topic, gene expression regulation. What does SRY do? It's a gene that encodes a protein. That's the central dogma. Hopefully everybody at least got that much out of the manual. So the gene is a DNA sequence. What's the first step in the central dogma? The gene encodes, there's another molecule other than DNA.
RNA.
Right, so DNA encodes RNA. We refer to that molecule as the transcript. It's another nucleic acid. It's just ribonucleic acid, not deoxyribonucleic acid. DNA, the double helix, encodes a molecule called RNA that we refer to as the transcript of the gene. And then what's the last step? We go from RNA to protein sequence. And usually, but not always, protein is the outcome of the central dogma. A gene encodes an RNA molecule. That RNA molecule encodes a protein. So the gene and the transcript, DNA and RNA, are related molecules. They're nucleic acids. They're long polymers of A's, T's, G's, and C's. And RNA, also nucleic acids, they're long strings of A, C, G, and U. There's a little difference. They're usually single-stranded, or they are single-stranded, not double-stranded. So we talk about a double helix of DNA. RNAs do sometimes base pair with themselves, but they're usually single-stranded, no complement. And then the protein is made up of what subunits? It's not nucleic acids anymore. Now we're talking amino acids. So this is the process we're going to be talking about over the next couple of weeks at least. It's the central component of genetics. Understanding how to read a chromosome sequence. And so I'm going to be asking you all to pretend like you're the enzymes that do these two steps. And what's the process of the first from gene to transcript? That process is called transcription. So I'm going to be asking each of you to take DNA sequences, transcribe, and to transcribe them. Pretend that you're the enzyme that does transcription. Here's a DNA sequence. If you were RNA polymerase, how would you read that DNA and what RNA molecule would you produce? That's transcription. And I'll also be doing the same thing, which is this, what's the second arrow? RNA to protein. The process is called translation. I'll be asking you the same thing. If I give you an RNA molecule, an RNA sequence, pretend that you're the ribosome and translate the RNA and tell me what protein gets produced, what the sequence of amino acids is. So we've got transcription and we've got translation. And those are performed by RNA polymerase, reads DNA and produces RNA, and the translation is done by a multi-protein complex called the ribosome. That's the big overview. How to take DNA sequence, chromosome sequence, figure out where the genes are, which is something that's really important for the cell because we've only got something like 30,000 genes in our genome. Most of the DNA in our genome doesn't do this process. Most of the DNA in our cells doesn't encode genes. It does other stuff. So one of the first tricks that people had to figure out, scientists, geneticists, and genomicists, after they sequenced chromosomes was trying to figure out where the genes are. So there could be, if this is an entire chromosome, there would be, in the humans, thousands of genes on this chromosome. But if every gene I encode is just a little box here, for example, there could be genes scattered all over the place. There are some genes that are longer, some genes that are shorter. Some genes overlap. Some genes are encoded on the top strand and some genes are encoded on the bottom strand. So remember, DNA is two strands of DNA. Some genes are encoded on the top strand, some genes on the bottom strand. Geneticists and genomicists had to figure all of this out. Where are the genes, and then what do they do? What are their transcripts, what are the proteins? Most of the time, the proteins are the things that do the functions of the cell, like transcription and translation. Ribosomes, RNA polymerase, DNA polymerase, and so many other proteins. So what I'm going to do, for most of the class, you saw there are a lot more slides than normal because I've included a lot of screenshots to walk you through the process. You should have seen the video of me doing this already, so I'm going to skip through this pretty quickly, but please stop me if you have questions. So how to figure out, the question was, what's the sequence of SRY? That was the first question to attack for today. How many nucleotides is the SRY gene? How many base pairs is it? So this is a URL you should all get to know, just for this class, and you'll use it in other classes as well, but this is a good place to start learning about NCBI, which is a component of the National Institutes of Health. This is a huge database that's full of all sorts of information that's really useful to biologists in general, but especially geneticists. It's the National Center for Biotechnology Information, which is what NCBI stands for. And it's inside the National Library of Medicine. So go figure. We put the databases inside a library organization. If you go to this website and you search for the SRY, so you just type SRY right there in the search field, you should come up with something that looks like this. If you don't, now, leave it at that for now. So a couple things. This is the database entry. It's like Wikipedia, except it's NLM. It's the National Center for Biotechnology Information. There are lots of different records. Here it's telling us the top hit is to the gene we're looking for, SRY. So you can poke one of two links. I'm going to suggest you choose the ref-seq gene first. That just stands for reference sequence. One of the issues with having a database full of DNA sequences is that I've got SRY, and wow, there are actually a lot of X individuals in the room today. So some of you that have Y chromosomes also have SRY. But your SRY sequence is not necessarily the same as my SRY sequence. So if yours was sequenced and mine was sequenced, we could both upload those sequences into the NCBI sequence database, and then there would be 2 entries for the SRY gene. And if we sequenced the same gene, which is in all primates, there's gorilla SRY, there's chimpanzee SRY, there's macaque SRY, bonobo SRY. There are lots of SRY gene sequences in this database from individuals or from different species. So this ref-seq gene is sort of the standard. It's like the definition of SRY. It's not one particular sequence, it's sort of a composite sequence, it's the average sequence, it's the reference sequence. So if you select that ref-seq gene link, you get taken to sort of this generic page. I think, yeah, so this is the ref-seq gene annotation. It tells you what chromosome the gene is on, there in the header. It tells you which species the gene is from, Homo sapiens. And it gives you this sort of brief history or information about the gene. So it tells you what the full name of the gene is, sex-determining region on the Y, which is what SRY stands for. And it gives you, for example, in this screen, what organism it's from. 2 things to point out here. One is a link to the FASTA sequence. So the question was, what's the sequence of this gene? This is how to do it. SRY into the search field, click on the gene name link, and click on FASTA. We've seen FASTA sequences before, that format. It'll show you what the sequence of this gene is. And you can copy and paste it and do all sorts of other analyses. The other thing that's important to note on this screen that I'll be asking you at least to know this term, the accession number. That's a unique identifier for every different database entry. So if there are 3 different SRY sequences in this database, they each have a unique accession number. Think of it like a call number for a library. Do libraries still have call numbers? They don't really have books anymore. Never mind, I'm old. So, unique identifier. Every individual DNA sequence entry in this database has a unique letter number combination thing. That's the accession number. So if I say, look up this accession number, you can type that accession number into the search field and hit enter, and it will take you to a single entry, whatever gene it is that's referenced by that accession number. Likewise, if I asked you to find a gene and tell me what the accession number is so that I can go look it up, that's what you would copy and paste or type or give as the response. That letter number combination is usually 2 letters followed by an underscore and then a number and then maybe some decimal places and other things. That whole line is the accession number. So when you select FASTA, here's the FASTA sequence of SRY. So if you were part of it anyway, because it keeps going down below the screen, it's pretty long. So there it is. That's the DNA sequence of the SRY gene, part of it. That's how you find the DNA sequence of a gene. Any questions? What might you do with the DNA sequence of a gene? What have we done with DNA sequences so far in this class? Think of your least favorite exam question. There was probably something that had to do with the DNA sequence in it. We've done sequence alignments. We've taken DNA sequences and aligned them together. That's one thing you can do if you have multiple versions, for example, of SRY. You could get all the real gene sequences of them. You could align them. I could ask you to look for some sort of a motif in here. How many times does the EcoR1 restriction enzyme site exist in this gene? Those are kind of inconsequential to do with the sequence of a gene. So if instead of hitting the FASTA button, if you go back a page, if you're following along right now, from that initial DNA, that initial gene record, if you scroll down, instead of going to ref-seq gene, you click on the other link and go down, it will give you other information which will be useful for us, like the translation. So it takes the DNA sequence, it says, here's the amino acid sequence in the protein that's produced by SRY. So this record, for every gene, not just for SRY, has a lot of useful information in it. Don't worry about the stuff that's above that. There's a lot of information here, but I will tell you when you need the information, if you need it. So we've got the protein sequence as well as the DNA sequence, just from looking at this record. And this is another format that's in that same long scrolling screen of, this is another version of the DNA sequence of SRY. So also SRY. Not in the FASTA format, though. So an overview of all the stuff you can get out of that sequence record from SRY in the database. Now, if instead of clicking on ref-seq gene, you clicked on that main link, that's where you would have gotten some of those pages we were just looking at, the additional information for SRY. And that would include a summary that you might actually be able to read and interpret. It's not just full of data, but this is something that some actual human wrote that's useful. This intronless gene, and that's one of the other things that's unique about SRY, or relatively rare about SRY. Not only is it just found on the Y chromosome, but once we get into transcription, we're going to talk about introns. This gene has no introns. And this also answers the next question. What is the role of this gene? So up here somewhere, you can look at it on your slides if you don't have it up on your web browser right now, if you're not following along, which is fine. What's the role of this protein? What does SRY, the protein, do? Inside the cell, what does it say up here? This intronless gene encodes a transcription factor. We're going to talk about transcription factors a lot in this section. Transcription factor is a protein that regulates whether or not genes are being transcribed or not. Transcription factor. It's a protein that tells genes when they're going to be turned on, transcribed, or not. Sometimes abbreviated TF. Shorthand, but transcription factor. Not surprising, because what does SRY do? What's its big function? What's the goal? What's the outcome of having SRY? You become, if you have it, you become male. So what SRY does is it's a protein that turns on other genes that are important in producing all the male traits, behaviors, phenotypes. SRY is the top gene in a big cascade. It's the switch. It turns on a lot of other genes that all together produce maleness. And that's why without SRY, none of those genes get turned on and no male, or few, male traits develop. This is unfortunate. This sort of idea has led to an unfortunate situation, or at least I think it's unfortunate. It's been debated for a while, the concept of a default sex. If you don't have SRY, what sex do you develop as? Female. And that's more of a socially awkward concept, I think, than biologically. It's true. If you don't have SRY, you won't develop male traits, because none of those genes that do the job of making you male get turned on if the switch, the master switch, if SRY isn't there. So in its absence, then the default is you don't develop as male, you develop as a female. Not entirely true, but that was an argument, especially in the 90s, that was made a lot in biology. So the concept is debatable, whether there really is a default sex. Okay, so that was all the questions and answers about SRY leading into this topic. Those are all of the things that we're going to be doing more in the next few days. Soon after the DNA sequence, or the DNA structure was identified, then people involved, like Francis Crick, had this idea about the central dogma based on a lot of different information, suggested that DNA sequence, and this is not correct, by the way, this is almost correct. It's an old idea. It's almost correct. That DNA encodes RNA encodes protein. So that's why Crick drew these arrows between DNA and RNA and protein in one direction. But wasn't really sure if sometimes DNA could directly encode proteins. That doesn't happen, to my knowledge. It might. It's very hard for biologists ever to make really strong assertions because there's always an exception to most rules. He drew these weird looping arrows, like, can DNA make more of itself? Does DNA replicate? Yes, but it involves proteins that replicate the DNA, like DNA polymerase. So DNA, it's kind of a chicken and the egg thing. DNA encodes a gene that encodes the protein that's necessary to make more of the DNA. Which came first? DNA polymerase or DNA? And likewise, wasn't sure if RNA could make more of itself, so drew this other looping arrow underneath RNA. But this is the really important thing. If there's anything important up here, he wrote, but never, and then underlined it, protein to DNA or protein to RNA or protein making more of itself. That is, the nucleic acid information is the code for protein. Protein doesn't encode DNA or RNA. Protein is the outcome of the central dogma, transcription and translation. But there are a couple of exceptions, and we'll talk about those. This is the general idea. So, Socratic. Let's see what the answers were. Question 1, does every gene, or question 2, does every gene obey the central dogma? Let's see, 49 of 53 of you answered. I'm not going to display it, but let's see, we had 63% false. So you can calculate how many said true. That is correct. Not every gene obeys the central dogma. There's an exception, or almost always an exception, and that's here on this slide. Why are we interested in genes? So this is just sort of a flowchart from a different textbook from a while ago, but it's still more or less accurate. Wow, it's actually, never mind, it's old. There are some genes on here that don't do central dogma. The main genes, when we think about genes, we're talking about coding DNA down in the lower left-hand corner. This chart says that, and this was a chart that was made before the sequence of the human genome, so this is, like I said, it's a little bit old, but it's still mostly accurate. About 10% of our DNA encodes genes that are transcribed and translated and turned into proteins. The other 90% of our DNA is what the rest of this is all about. What is the rest of that 90%? What does it do? Why do we have it? What's its function? Why do we bother replicating it all every time every one of our cells divides? So where on here are genes that don't get translated? They don't go from RNA to protein. Over here, there are 2 RNA genes and 22 tRNA genes. Those are genes that are meant to be RNA. That is the output of that gene. It gets transcribed, an RNA is made, and that's the product, is the RNA. Those genes never get translated into protein. So those are the key exceptions. Lots of genes just encode RNAs. The RNA is the functional product of the gene. And we're going to talk a lot about tRNAs when we get to translation, because those are the RNAs that bring amino acids to the ribosome for synthesis of proteins. So in terms of tasks for this topic and vocabulary. A lot of this vocabulary is introduction to all of the transcription and translation vocabulary and some bioinformatics things, like BLAST, query, and subject. We've done one of these tasks already. Maybe I skipped forward. We've done restriction, fragment, length, polymorphism, agarose gels. That was the last topic. So you know how to do that, or you have started to learn how to do that. We'll continue practicing it, but that's not one of the main focuses of this topic. We're going to be focusing mainly on the bottom four. We're always working on that second one, evaluating experimental conclusions. And this topic, again, we're going to turn back to a case study paper. So for next class, one of the topics, one of the things to do is to go read this paper. There are a few questions about it in the course manual. We'll talk about those next class. So that's where we're headed. It's mostly DNA sequence information analysis. BLAST is what we're going to talk about right now, and then eventually proteins. One of the other Socratic questions was, what's the misspelling? What's the percent identity of the two gene sequences? That should be a T. So what is percent identity? Most of you got this. Some of you said I didn't know. That was fine. We haven't done this yet.
How close they are to being the same.
Right, percent similarity, how close they are. So this is an alignment. What's the process of calculating a percent identical? What fraction of the nucleotides are the same? So if you're going to do that, what do? These are total of 20 nucleotides. We look for SNPs, or any sort of difference between the two sequences. So how many are there? There's one there, one there. Is there another one? One there. I think that's it. Is that right?
Yep.
3 differences? So 3 out of 20, that's differences. So identity, how many nucleotides are identical? Top strand, second strand, first strand, second strand. How many are identical? 17 out of 20 identical nucleotides. And if you translate that to percentage, it's 85%. So 85% ID, 85% identical. And this is a way that geneticists assess how closely related gene sequences are. Line them up. How similar are they? Turn it into a number. So there were lots of 85%. So some of you guessed, some of you didn't, about half the class, 85%. Plenty of time to work on this in the future. So that's the concept of percent identity. If you have 2 DNA sequences, you line them up, you just count how many nucleotides are the same, identical, out of the total, the fraction, the percentage. That's the answer. So take 3 minutes, maybe. Don't want to give you too much time on this one, because this is a misleading question. Trick question. Down here, there's a sequence of something I made up, and it's aligned with 3 other sequences. So the question is, compared to this top sequence, is number 1, number 2, or number 3 most closely related to the top sequence? I really hope you will do this and talk with somebody if you've got any questions. Come back together in 2 or 3 minutes or until discussion stops. Which is the least closely related? How many differences are there between the top sequence and sequence number 2? So differences 2. So what's this percent identity? I think I wrote it down here. 13 out of, that's 15 long. So 13, 15 is, yeah, about 87%. 87% identical. What about the comparison of the top sequence and number 1? Are there any differences? If they were identical, then they would definitely be most closely related sequences. So there's one difference. What's the percent identity between 1 and 2? Bless you. 14 out of 15? 93% identical. So what's the last one we have to do? Comparison with number 3. How many identical nucleotides?
14.
Also 14. There's one, where's the difference? Oh, right here. Yep, the same place there was a gap, a missing nucleotide in sequence 1. So that G is also a G in sequence 2, but there's nothing there. There's been a nucleotide deletion in sequence 1. There's a different nucleotide there in sequence 3. So its percent identity is all what?
93.
Also 93%. 14 out of 15 nucleotides are the same. So what's the answer? Which of the 3 is most closely related? Good question. Can you have 2 answers? Is this one of those situations where there could be more than one right answer? What's the essential question to ask, do you think, what's the difference between 1 and 3?
One's a deletion.
One's a deletion, one's a SNP. Is there a difference to a geneticist? Is it, say, more or less similar to have a point mutation than it is to have an insertion or deletion? Which is more common? Maybe the more common type of mutation counts more towards being similar or identical than the other one. This is a conversation that geneticists do not have an answer to. So you're right, in this case, you would say both of those are equally distantly or equally similarly related to the top sequence because they do have the same percent identity. But geneticists disagree on whether or not gaps, deletions or insertions, or point mutations should be more or less heavily considered when you do this sort of a comparison. And that's important when we start talking about BLAST. BLAST is the most commonly used genetics computer tool ever. It's really powerful, it has a lot of uses, it's free. You also access it on the NCBI website. Look, here it is. Another URL, but it's the same base URL, the National Center for Biotechnology Information. It's just like any other search tool, but this is a search tool for DNA sequences. Instead of pasting in a search term, you paste in a DNA sequence. And then BLAST, this algorithm, program, the computer tool, searches the sequence database and looks for matches between whatever DNA sequence you told it to look for and all of the millions of nucleotides that are in the DNA sequence database. So this was another exercise to have at the start of class, or for the start of class. This is one of the exercises. How are the 3 sequences related to each other? Or are they related? A, B, and C in FASTA format. Are they related to each other? Do they have anything in common?
Yeah.
They're all identical, 100% identity. Same there. So basically it's a short, a medium, and a long version of one gene sequence. So, did you blast those 3 as the exercise suggested? And if so, what did you find? What gene is this DNA sequence from? So if you copy and paste that DNA sequence, any one of those 3, what gene is it? So this is what what it should look like today, anyway. Changes quite frequently when you go to that NCBI BLAST website. You want nucleotide BLAST. Nucleotide, DNA. Don't want protein BLAST, that's that other big button. We're not at proteins yet. So nucleotide BLAST. Hit that. You just paste in that sequence. Use the FASTA sequence or not, it doesn't matter. You can either leave in the greater than A, the header, or not. You can just paste in a DNA sequence with no gene name in it, that's fine. And then that BLAST button down at the bottom left. That's the program taking that DNA sequence and just matching it against every DNA sequence in the entire database and looking for matches. It's going to give you the best matches. That's the result page that's going to come up next, which looks something like this at the top. And each of these is a link to one of the results. It's like Google search results, top down. The top one is the best match to your sequence query. And you'll note over here, for every one of these matches, it gives you the name of the DNA, the name of the organism usually that the sequence is from. In this case, Stenotrophomonas nitridoreducens strain 2001 something. Sounds like a bacteria. That's not a gene. We're looking for the name of the gene that this sequence is from. And by the way, this is the short version. That was sequence A, the 20 nucleotide chunk of this DNA sequence. Over here on the right side, the different columns give you different information about how good the search result is. That is, how closely that record in the database matches the DNA you paste in. And in this case, what's the percent identity in the top hit? Maximum score 42.1, total score 102. You don't need to know those things. Percent identity, 100%. Perfect match. Okay, how many 100% matches are there? All of them in this screen, and then keep going down. Right? There are a lot of DNA sequences in this database that are a perfect match to those 20 nucleotides. And what species are they from? We've got, so Homo sapiens, Homo, human. Cautoperca gobio, sounds like a fish. I'll wager anybody I haven't looked it up, but it sounds like a fish species name. And then these ones at the top, bacteria, Stenotrophomonas. We don't even know, at this point, we don't even know what species this gene is from, much less what the name of the gene is. If you scroll down even farther, you get to see the actual comparison, the pairwise alignment of the DNA sequences with the matches in the database. So this is the important terminology. Query is what BLAST calls the sequence you input. It's what you're inquiring about. It's the query sequence. The subject sequence is the database entry, what the query sequence is being compared to. So in this case, the database entry is this Stenotrophomonas complete genome. So in this bacteria's genome, it has here, subject, between nucleotide 2,691,352 and 2,691,332, that's what those numbers are on the left and the right sides of the alignment, has a perfect match, 100% identity, right there, 21 out of 21. Oh, 21? I thought it was 20 nucleotides. Well, I guess I was wrong. 21 nucleotide sequence matches from the start, the first nucleotide of our sequence that we pasted, to the end. Perfect match. Bacterial genome. So that's what all of these bits are. Query sequence, subject sequence, and then also we have the percent identity calculated for us. And you can see the actual alignment of the nucleotides. So is this a gene from a bacterium? Or what's underneath this alignment? What's the next best match in the database? Homo sapiens, opsin. It's a gene that's in your eye that lets you absorb wavelengths of light and see in color. What's its percent identity? Also 100%. So how are we supposed to figure out which which organism this was, much less what gene it's from. Is this a bacterial sequence that I gave you, or is it a human sequence? Or is it something else? Because these are just the first two of many 100% matches. So if you skip down to the longest sequence, which I thought was 40 nucleotides, but maybe it's 41, I don't know. It's about 40 nucleotides. And you do the exact same thing. You paste it into the blast window, you get the results. This is what you should see. What's different between this version of this window and the one we just looked at? There are 10 sequences, and where did the bacterial sequences go? They're not there anymore. The bacterial genome sequences are no longer the best match. Now we have, what are these sequences? They're from human, 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 chimp, chimp, and pongo, orangutan. And now all of these are saying they're long wave sensitive opsin, 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 opsin. What's different? Oh, by the way, here are those same alignments. So we can see, for example, that the human, our DNA sequence, the 40 nucleotide version, 41, I guess, is a perfect match to which sequence is this on the top? Subject sequence, that's the database entry from humans. Here's Pan troglodytes, non-human primate. What's its percent identity? 98. Where is there a difference?
The third nucleotide.
Right, so this is human has a 100% match. This, whatever sequence this is, it's from human. This was the gene sequence of human opsin, long wavelength sensitive. It's the one that lets us observe long wavelengths or red light. So this is the human long wavelength opsin sequence, and it's got a slight difference when you look at the same gene in a closely related species. So this is why you should, in future, conclude that this is indeed a human gene and not the chimpanzee or orangutan version of the gene. The higher percent identity is the best thing. Except for that pesky thing about those bacterial genomes, because they also had 100% matches. So how do you know that this is a human gene and not a bacterial gene? So last point. Thanks for hanging out for a minute if you're interested. Why did these results differ depending on whether or not we used the 21 or the 41 nucleotide long query sequence? Based in a little bit of the gene versus more of the gene.
It's more specific.
Because
I don't know how to say it. There's like more that you have to get right.
there's more information.
Yeah.
We've talked about this a lot. The probability of having one nucleotide, A, G, T, or C at any one spot, plus the combination of 2, combination of 3, combination of 4. A 20 nucleotide sequence is found much more frequently by chance than a 40 base pair long sequence, where you have to get every single nucleotide right in a row 40 times. That's why I put down here at the bottom, I just checked today, that database that we were just looking at today has 57 million DNA sequence entries. The total number of nucleotides in that database right now, it's 258 billion. There are 258 billion letters every time you hit blast search, it searches your query against. And that's why it's important to use the longest sequence possible to get the most precise results. So do what you have to do for next class, right up here, and I'll see you on Monday. Thanks a lot. Have a good weekend.